It's 1 p.m. in Los Angeles on Tuesday, the 7th of August, 2012. I'm Mark Strassman, reporter with Utopia News. I'm about to speak with Joachim Messing, who is the University Professor of Molecular Biology and Selman A. Waksman Chair in Molecular Genetics at Rutgers, the State University of New Jersey, and the Director of the Waksman Institute of Microbiology. Welcome to Utopia News.
Thank you.
We're going to talk about a plant called Spirodela polyrhiza, or in English, duckweed. We're going to talk about it in sort of its macro qualities, and then we're going to talk about it from its genetic perspective. And I wanted to start by asking you what makes Spirodela polyrhiza, or in English, duckweed, so special?
Well, it's the fastest growing plant on Earth. And if you try to capture photosynthesis in terms of biomass, it has a lot of attraction.
Okay, and where does it grow?
In nearly all geographic locations and altitudes, and it requires water. It floats on water, and many people, I think, in the States can see it if they walk along some low-flowing streams where there is a green carpet on the water. That's usually duckweed. They're very small, tiny leaves. So there are other aquatic plants, but these are very small, tiny leaves that float on water.
You said it's the fastest reproducing plant there is. How fast does it reproduce?
Well, it's, if you have optimal conditions, I think it doubles every couple of days. So you could cover the whole planet, if you had enough water and of course, nutrients for it.
Okay, and what role does it play in nature now? Where does it fit into its ecosystem?
Well, it has spread out mainly because of utilizing a lot of the nutrients in standing waters. So, for instance, it is well known for taking up nitrogen and phosphorus. So that is something that is generated, for instance, in suburban areas from using fertilizers for the lawn. If you put your fertilizers on the lawn, there's an excess of nitrogen and phosphorus that gets into the streams. And they like that, these duckweeds. And the Environmental Protection Agency uses it to measure water contamination.
So what's its value then in environmental remediation? How can this ability to uptake phosphorus and nitrogen be used to clean water?
Yes, I think in some parts of the world it is used for treating municipal water. They just put it out there to clean the water. Of course, there's some additional cleaning required, but it absorbs, it takes up nitrogen, phosphorus, and I think this is mainly in, New Zealand and Australia. I'm not so much aware of whether local communities here have already picked up on it.
Can it be? Is it used for food by animals in the wild, and could it be used for animal feed or even for human consumption?
It is. It is used for as a protein source. It has well-balanced protein, and it is used to grind a powder, which is then used to supplement food and feed, but not in this country. This is in overseas countries.
And what's its potential as a feedstock for biofuels?
Well, I think it has great potential, and here in our neighborhood, there is a startup that is demonstrating its use as for making electricity. And what is one of the great things here is that it is, in contrast to any other biomass, it is neutral in terms of CO2 generation. Why? I explain it to you. So you grow it on water, and what happens, the pH of the water rises, the other way around. So the water gets alkaline. You understand that?
Yes.
Yes. So when it's harvested and it put in a combustion oven to generate energy for a generator, you can capture the CO2 that is generated because of you burning carbon, right?
Right.
And that CO2 is put back in the water to lower the pH. So then it can start growing again.
Good. So
you recycle the CO2.
Good. What biofuels can it be used for?
Well, as I said, this example, this company here in southern Jersey is using it to produce electricity, and I think they have already a contract with PSE&G here. That's the power company in New Jersey, okay? And so the trick here is for this development to recapture the value, PSE&G uses them for carbon credits. But the electricity is, mixed into the electricity that they distribute. And, for the carbon credits that PSE&G gets, because they're using fuels that generate CO2, they can, make it profitable for using duckweed as a source for electricity.
So it fits into the economic system as well as the ecological niche that it fills.
Yes, there are other uses. What's happened in the life cycle is that when it gets cold, of course the water would freeze and it would kill them. So what they do is they switch to a different developmental stage, and they create structures that are called turions, T-U-R-I-O-N, turions. And what it is full of starch. The starch content in these turions, these are sugars, starch.
Right.
Starch. It's getting up to 60% starch, and that makes it heavy. So it falls to the ground, and then the top can freeze, but the ground is still okay, and they survive. In the spring, when the water warms up, they return to normal leaf structure, and they're using the starch to, as an energy source to come up again. But, one could harvest these turions, and one would have a source of starch. Like now, starch is used for fermentation. As you know, it's a bad thing that they use corn starch for gasoline or blending gasoline.
So this could be a source of ethanol as well?
So you could, this would be probably, this doesn't compete with corn as a food crop. So I think as an alternative, this would be much better. Now, let me give you a general comment about biofuels.
Okay.
Big problem with biofuels is the cost of transportation. If you make electricity or gas or so on, to take from one place to another, that's too expensive. I think an ideal model would be to develop this for local communities and not, ship it here from the East Coast to the West Coast. So, what I said about, remember, I said I think it's in New Zealand where they're using municipalities to clean up the water. So you can envision local communities use it to clean up the water and then they harvest it. And, it could be used, as I said, either for electricity or for fermentation. But the importance is that this process of harvesting, okay, is very important because it's not so easy to separate the water. See, this is the major challenge with algae. You may have heard of microalgae as the future, right?
Right.
But the problem is they are so small, the algae, that you can't get rid of the water easily. It's expensive to do that. The duckweed, the leaves are large enough that separating the water is no problem.
All right, so I was going to ask you about how it compares with algae, and what you've said is it's a lot more efficiently harvested.
Yes.
Okay, I want to turn now to the internal structure, the genome of duckweed. When did you start working with it?
As I said, the duckweeds go everywhere, and there are many different. There are 5 genuses, as we say. So Spirodela, then there are other ones, and and within all these 5 genii, there are also different species, and within the species you have different ecotypes. So there is a scientist in Zurich, in Switzerland, Landolt. He has been collecting these, and he had a good collection. So what we did here is we looked at the collection and tried to find a species that had a small genome.
Right.
So the reason for this is if you want to sequence one of them, you try to find one with the smallest genome, okay?
Right.
From the 100 different samples, ecotypes, species, and different genera, I think you say plural. The Spirodela had close to the smallest genome size, and it was about, I think, 160 million base pairs. That sounds like a lot, I know, but the human genome is about 3 billion, so it's about 5% of the human genome. But if you take, for instance, maize, a corn, that has a size like the human genome, a little bit smaller, but among the plants, flowering plants, I think this is one of the smallest genome, besides duckweed, which besides Arabidopsis, which is a weed that has a small genome, and people that worked on it a lot.
Is there a connection between the fact that it has such a simple genome and the fact that it's the smallest and fastest flowering plant?
No, I don't necessarily think that would be too early. So you're calling me at a time where we have yet to analyze the gene set of it. The sequencing has been completed. But what will be necessary to do, and you can call me again when we're done, is to make a comparison of the gene set of Spirodela with other plants and do some sort of subtraction to see, what genes are, particular active in Spirodela, and see how it matches with the morphogenesis. What I mean by that is, it doesn't have really a stem like other plants, so it doesn't need any support structure. It just floats on water. So it probably, I guess now, I don't know, it probably has not a lot of lignin synthesis or cellulose synthesis, which you have, of course, in other plants, important to get all the support structure.
Because it's simple, it doesn't need that much genetic information to make itself.
We don't know that yet. In many cases, in flowering plants, the minimum gene set I think is close to 9,000 genes or so. But what's different is the amplification of genes. So you have, let's say, one gene, and then you get several copies of it, right?
Right.
What happens a lot is nature is very inventive and innovative. It uses the genes all over again, but different times, or they can switch them around for different uses. And so what will be an interesting aspect to see what genes are amplified in Spirodela as compared to, for instance, to rice or banana or tomato. See, we have now quite a few genomes that we can compare it to, so we get a good idea of the evolution of these aquatic plants.
Tell us a little bit about the process under which duckweed was adopted by the U.S. Department of Energy's Joint Genome Institute and the Community Sequencing Program, which gave you access to more powerful sequencing tools.
Well, the Department of Energy had developed a central laboratory that has a capacity to sequence DNA, any DNA that you send them. And what you do is you send in a proposal and say, hey, take my DNA. And the reason you should take my DNA is because it is close to your mission. Your mission is to identify the genetic information of organisms that have potential as an energy source. See, I had done with them a couple of genomes, like sorghum is another potential biofuel species, and that has been done the same way. They look what capacity we have this year, and they queue in, from all the labs over the country. They send them the DNA, they sequence that DNA, and actually it's more volunteer work than to analyze the data. You don't really have any money for that, and you don't get any money from them. They just have the machines, and they can buy the reagents to sequence your DNA.
Now, what will having part or all of the DNA decoded do to help you modify the plant to emphasize various aspects of its characteristics?
Yeah, I mean, we have already taken some steps before all the genes were known. Like we have looked to I told you about this season switch.
Right.
Forward to the bottom.
To make the turians.
Yeah. So we can induce that in the laboratory, and we have looked for one particular gene family, and that is the entry step to making starch. Of course, the reason for doing that is to find promoters that would be specific for turion turn on. So one thing you can envision, if we know all the genes and so on, that we might make new combinations with promoters and genes. Maybe we want to increase something else than sugar, maybe oil also. which would be very different, but you know, there are different interests in what source of raw material would be desirable, you know. Of course, starch is useful for fermentation, but you could also generate other macromolecules that are of interest.
What's the time frame for the development of some of these new applications?
I think in the next 5 years you will see, you know, customization for the different applications. You see, like this local company, I think if this takes off, you know, I think there will be, you know, if they have, let's say, a process here that will generate electricity, there will be probably like to make, let's say, 10% improvement. You measure it somewhere, you know, and of course, if you sell, let's say, $100 million of electricity, and you can make 10% improvement, that's 10 million. That's worthwhile, I think.
Right. Now, who's going to organize and possibly profit from the genetic modifications? Who owns these genomes, if anyone?
No, the genomes are not owned. I don't know how familiar are you with this area of patenting.
Not too. Give us a brief introduction.
So, I think there was a major ruling that you can't patent a gene.
Okay.
You only can patent an application of a gene where you know what the particular function would be. So, so if we, let's say, sequence the whole genome, the genes are all free. You know, if you find something out that a particular gene is good for something, then of course you can file.
Okay.
Actually.
Okay. Well, finally, what do you see as the future of duckweed, and what role would you like to play in its development?
Well, I'm just a you know, professor here at the university. I have. You know, I have over my career, I have done both. I have, you know, focused on basic research, and in this case, I think it will be interesting to understand a little bit more about monocots and the evolution of these species. But I also have had always an interest in universities being an economic engine and creating new jobs for the local economy. So we will have a lookout for partners in industry that are interested in this and see how we can help them, you know, to transfer the knowledge into practical applications.
Okay, good. Well, I want to thank you very much for your research and for talking to us about it today on Utopia News.
You're most welcome, Mark. Okay, you take care.
You too. Bye.